Hello, in this video we will discuss about the epigenetic changes, histone modification, and we will learn about and how the DNA silence, the gene silence and active the gene to produce the product. So let's begin to understand. First of all, the epigenetic is the changes of gene regulation without changes of genes. It means the nitrogenous base will not change, the DNA segment will not change, but the changes due to the addition or addition of acetyl group or methyl group, and histone acetylation, histone methylation, histone phosphorylation, histone ubiquitination. This is the four type of addition of the extra molecule in the DNA to change their function. For example, silence or express. So this is the tissue of the person which that contain a nucleus, cytoplasm, cell membrane and nuclear membrane, and inside the nucleus is chromatids. This is known as the chromosome in the form of chromosome. So let's draw a chromosome structure. This is the chromosome structure is basically contains sister chromatids which that contain a allele which on the locus. So the different loci contain different type of alleles, the genes, active or inactive. In middle is the centromere, and the diameter is 1040 nanometer. 1400 nanometer chromosome. While here is the tetrameric, the tetramer histone protein in the form of 8 subunit of histone protein with the type of H2B and H2A lie together and H4 and H3. So this is known as the nucleosome. It is the repeating unit in the chromosome which that contain a DNA as well as protein. Histone protein is 60%, DNA is 40%. This is the DNA after all, which that is the genetic material. Contain nitrogenous base, adenine, thymine, guanine and cytosine. So the antiparar adenine always bond with the thymine and guanine with cytosine through hydrogen bonding, and this is the CpG island methylation. CpG methylation mean the 5' to 3', and the cytosine and guanine rich content are present in the DNA when. So antiparar strand. So let's begin to understand the histone modification with the diagram to understand methylation and acetylation. What is this? So we will focus on the acetylation and methylation of histone. Not we will learn about the phosphorylation and ubiquitination. So this is the linker protein also present, the histone 1, while the H3, H4 and H2A and B are present together, and the tetramer. So here is the methyl group attached with the histone tail, and the acetyl group attached by the hat enzyme. So the hat enzyme basically demethylation. So the methylation to inactivation will lead to demethylation. Here is the linker protein, and in this way here is the acetylation will occur. The acetyl group will be added while the methyl group will be removed through a hat enzyme, and in this way the HDAC basically is the deacetyl enzyme, deacetylase enzyme basically will lead to remove the hydroxyl group, acetyl group will lead to, here is the histone acetyl transferase hat is uncoiling of DNA used because the acetyl transferase will lead to acetyl group addition, while the histone deacetylase enzyme HDAC is used for the coiling of DNA because the deacetylation to add the methylation group for the gene silence. So whenever the acetyl group will be added, the gene will be expressed in the form of production of protein. So the best known modification of terminal histone region is methylation of lysine 9 at histone H3K9, which in mammals for gene silence. If if gene acetylation abnormally lead to cause cancer. So remember these things. This is the important thing you should need to understand. So anyhow, the acetyl group is used for the gene expression, while the methyl group is used for the DNA silence of the gene. So here is the mechanism. The transcription will be occur when acetyl group will be added. So the antiparallel strand are present. So the basically the template are 3 prime to 5 prime RNA is the basically is the copy of RNA will produce. This RNA is the messenger RNA because get the message to the cytoplasm, and where the cytoplasm the translation will occur to produce when ribosome will bind to produce different type of protein and different protein form, and after this maturation through a Golgi bodies and endoplasmic reticulum, and in this way the different type of protein, functional protein and anabolic protein, the channel creation, the channel aquaporine like protein, a porine protein used for the active and passive transport of the content, while the enzyme producing gene, enzyme produce channels as well as the receptors, anabolic protein and membranous protein which that is produced. Even the whole cell will be controlled by the production of protein for the homeostasis and maintenance of the cell. So the interplay between DNA methylation gene transcription, let's begin to understand how the transcribe of the gene into the protein. So let's begin to understand. First of all, we will learn about here is the active open chromatin and the condensed closed chromatin. We will learn about these things. So this is the histone protein tetramerically bind with the 4 type of histone protein H2A, H2B and H3 and H4 and linker protein H1. In this way here you can see the unmethylated cytosine which that contain acetyl group obviously is used for the transcription will lead to produce the protein. So the methyl group will balance should be when the methyl group is when the requirement will be not for the production of protein, so the methyl group will be add partially but remove when the requirement of the synthesis of protein. That mechanism is also involved for this function. And the methylated cytosine, basically methylated cytosine will silence the gene off, and the transcription will not occur. So remember, the methyl group add when the requirement of protein will not. When the requirement of the protein will occur, so the acetyl group will be activate the uncoiling of the histone protein DNA wrapping around the histone protein. Uncoiling will lead to transcribe and translate the things and the proteins. So let's begin to understand the DNA methylation, how it's possible. DNA methyl transferase 1, 3A, 3B is involved, that enzyme, and the SAM mean S adenosyl 1 methionine, which that will be add. And in this way you can see after remove it, basically it is as a substrate working and due to the presence of the enzyme will lead to transfer the methyl group. After the methyl group transfer by the removing of the proton, so here so in this way the 5 methyl cytosine will be formed after the methylation. So it is known as the cytosine methylation, 5 methyl cytosine. While the CpG island are present in the promoter region mostly 5 prime to 3 prime, here is the DNA structure which that contains CpG. This P mean the phosphate group are between the cytosine and guanine with the cytosine antiparallel. And in this way the methylated DNA, this for example in the promoter region will lead to methylated. So unmethylation will occur when the acetyl transferase enzyme will work. The histone acetyl transferase enzyme HAT, which that will lead to uncoiling of DNA, will lead to histone deacetylase will again coiling. So it in this way the transcription will occur to translation. But the CpG island are present in mostly promoter region of the DNA will lead to promote the natural gene, while the promoter is used and control the regulation of the gene transcribing. So the CpG methylation will inhibit the natural gene, while the oncogene, for example, will enhance due to the promoter. CpG acetylation will extra hyperacetylation will lead to activation more oncogene, will lead to cause cancer. So that is why the epigenetic changes and histone modification will maintain the body cell for the prevention of the disease, for the prevention of rapid aging or something. So the cytosine phosphate guanine, the CpG or methylation will occur by this mechanism, DNA methyltransferase 1. So I hope you make sense. This video about the epigenetic changes, histone modification, and how histone will be modified, for example acetyl group add or methyl group add to regulate the gene. And this is the epigenetic video about the histone modification and the epigenetic changes. So thanks for watching. Please make sure to subscribe, like and share. If you want to get more video about the epigenetic, check the playlist to understand more process. So I will make another video about the DNA methylation and relation with the cancer as well as other disease. So